Lorena.
Well, thank you, Jackie, for that introduction, and thank you all for inviting me here today. What I'm going to be talking today is a little bit of the work that I've been doing as a postdoc in the lab of Joel Sachs at UCR. And so I don't know if you all know, but specialization in species interactions is thought to be one of the main drivers of diversification. So just as an example, orchids is the second largest family of angiosperms, and they have developed these specific structures in their flowers to attract pollinators. And so these specific interaction might be driving the diversification in orchids. It's almost 25,000 species. Another example of that is the case of the Fabaceae, which have evolved a very specific interaction with soil bacteria called rhizobia. Legumes tend to associate with very specific rhizobial lineages that you can see up here. And it is thought to be a major driver of the different speciation events happening in legumes. So those great lupins that were shown yesterday, they can potentially be speciating because of these interactions with the soil rhizobia. And so that's what I'm really interested in understanding, how these associations, or these symbiotic associations, can actually lead to diversification. And the processes are not well understood just yet. So for those of you that are not familiar with the legume rhizobial mutualism, that you can see here the gray lupin, they tend to associate with the rhizobia in exchange for nitrogen. So the plant gives carbon to the bacteria, and the bacteria gives nitrogen to the plant. And they tend to form those structures in the roots called nodules. How can we actually measure that level of specificity between the bacteria and the legumes? So one way that we can do that is by measuring three different traits. When you expose legumes to specific rhizobial lineages, you can measure the ability to form nodules or not. That's one specific trait. The other trait that you can look at is how effective was that infection. And one way of doing that is by counting how many nodules were actually formed. And I forgot to say that bacteria actually live inside these nodules until the plant flowers. Once the plant flowers, they are released into the soil. And more importantly, we need to measure if this association is actually good or not. And we need to measure the growth benefit. And one way that we do that is by comparing the growth of the plant with the rhizobia compared to the growth of the plant without the rhizobia. And that's what we call growth benefits. But I'm not focused necessarily on lupins. I am very interested in a native genus to Western North America, called Agnesmon. It used to be called lotus, and recently it was separated between the New World and the Old World. The Old World are called lotuses, and the New World are called Agnesmon, among other genera. Just in California, there are like about 23 species and more than 36 varieties. And one thing that really upsets me about this genus is how many different species are adapted to really extreme environments. You can find them in the deserts, you can find them in serpentine outcrops, but more importantly, you can find them in nutrient-poor soils and usually in very disturbed areas. Also, from what we know is that there has been different transitions in the lineages that they are associated with of the rhizobia. So far, we know that the most ancestral Lotus japonicus, one of the Old World, tend to associate exclusively with Myxorhizobia. But Agnesmon species tend to associate mostly with Bradyrhizobium, except for this transition here between Agnesmon strigosus and Agnesmon magellanus. What is interesting is that these two species are usually found in nature in the same habitats. You can find them right next to each other, and they tend to have a sympathetic distribution. So it's really interesting to think about it in terms of like, is this transition leading to speciation or not, or how it happened, why did it happen? And that's the main focus of this little project that I'm showing you today. But just to let you know, out of the 23 species, we only know the symbiotes for these 5 different species. And part of the bigger job that I'm doing right now is trying to understand, first of all, reconstructing the phylogeny of agnusperm and trying to find what are the symbionts in each of the different species to understand how many switches have happened across the genus. And so stay tuned for that. We just got some genome skimming data along 300 genomes of the bacteria symbionts across the different species. So I have 2 different hypotheses about this transition. One is that a more generalized host evolved to become specialized, and if that is the case, both species should retain the ability to associate with the alternative lineage. An alternative hypothesis is just that a switch happened between these 2 species really rapidly. And if that is the case, we should see that they are both highly specialized to their own symbiont. And so they shouldn't be able to associate with a heterospecific lineage of rhizobia. So pretty much what I wanted to test was whether Agnusperm strigosus and Activisperm granularianus retain that ability to associate with a heterospecific lineage of rhizobia or not, and how specific they are to their own rhizobia lineage. And ultimately, try to understand if there is genetic variation in these specificity traits that could potentially lead to rapid evolution to associate with alternative lineages. So we have used different genotypes collected across the range of both species. You can see here, this is data from the GPF, where one of the species tend to be mostly distributed in the northern part of California, and that's Activisperm granularianus, and Activisperm strigosus tends to be mostly distributed to the southern part. We collected different genotypes across the different populations, and just to let you know, we also have collected the rhizobia from these natural populations from both species. So we have isolated bacterial strains that we can actually test and inoculate these different genotypes. So pretty much what we did was a reciprocal inoculation experiment where we inoculated the genotypes of strigosus and granularianus with 2 different strains of bradyrhizobia, and also with 2 different strains of Mesorhizobia. In addition to that, we also wanted to know what happened in nature, what happened when they are exposed to both types of rhizobia. And that's what we call co-inoculations. And pretty much we use one single bacteria of the bradyrhizobia in combination with one of the strains of the Mesorhizobia. And we did the same, but only with the meso. So we only changed one of the bacterial strains of the Mesorhizobia. It wasn't a full cross of all 4 strains. So this is how the experiment looked like in the greenhouse. We have 5 replicates for each of the genotypes, and pretty much we ended up having 7 treatments because we also had a control where the genotypes were not exposed to any rhizobia. We only added water. And after 6 weeks post-inoculation, right before they started flowering, that's when we did the harvest of the experiment. We pretty much split the biomass, the above ground and the below ground biomass. Then we have to separate and count each of the nodules, and randomly selected about 5 nodules per each plant that we have to sulfur sterilize, grind, and then do serial dilutions to then be able to plate them and do a count of how many bacteria were present in each of the nodules, and also to identify if they were being colonized by Mesorhizobia or bradyrhizobia. So what did we find? What you see here is the different strains. So we have two bradyrhizoian strains, two mesorhizoid strains, and here you're going to see the trait of interest. In this case, is the probability of nodulation. Was it able to form nodules or not in the presence of that particular rhizobium? So for Strigosus, for Agnuscon strigosus, what you can see here is that they are very specific to their own bradyrhizobia. They tend to have higher probabilities of forming nodules with their own conspecific lineage rather than with the heterospecific lineage. The same happened for Agnuscon granularius. And so some genotypes were able to actually form nodules. Now the question is, how successful was that infection? Well, it wasn't that successful. Again, we see that with the conspecific lineage, they tend to have higher number of nodules than with the heterospecific lineage. Same happened for Agnuscon granularius. So the take-home message from this is that both species retain little to no ability to associate with the heterospecific lineage. They are highly specific to their own rhizobial lineage. So now, with those few genotypes that were actually forming nodules, we also wanted to know were they getting any benefits. Was this association actually successful or not? What we found is that it was really not that successful. In the case of Agnuscon strigosus, they were significantly different depending on the strain. But that was not necessarily the case for Agnuscon granularius, where statistically speaking, so it seems that, yes, they're highly specific, but it seems to be as asymmetric, where Agnuscon granularius might retain some kind of generalism. And this is the canoculation experiment. For this is the data from each of the nodules that I was telling you about, where I harvest a lot of the different bacteria. So we wanted to see were they actually able to associate or not with their rhizobial lineage or not. And what you can see here is the different genotypes of Agnuscon granularius, different genotypes of strigosus, and they tend to be, again, very specific when both strains are present. They tend to select one or the other. Now, in terms of the genetic variation for these different traits, what you see here is the mean trait value for each of the genotypes. And the spread of these points is going to tell us how much genetic variation there is for that specific trait when they're inoculated with that specific lineage. And what you can see here is that for Strigosus, as we were expecting, there is a lot of genetic variation for associating with bradyrhizobian strains, not so much with Mesorrhizobia. The same pattern was observed for Agnuscon granularius, but in this case, actually we see some genetic variation to associate with the heterospecific lineage, and not so much with its own Mesorrhizobial lineage. So there are some genetic variation in Agnuscon granularius, but not so much in Agnuscon strigosus. Now, in terms of the number of nodules, we observe the same pattern for Agnuscon strigosus. And what is interesting here is that for Agnuscon granularius now, we're seeing genetic variation, but actually we're seeing a genotype by rhizobial strain interaction, which is telling us that there is genetic variation in plasticity for the different association with alternative lineages of rhizobia. So in conclusion, both species tend to be highly specific. They really retain very little ability to associate with alternative lineage, with Agnuscon granularius showing some ability to do so. And pretty much we see some segregating variation in nature for associating with conspecific lineages, and in Agnuscon granularius we see some variation for actually be able to rapidly associate with a heterospecific lineage. So coming back to my original hypothesis, I think our data is really strongly suggesting that the switches happen really fast. This is still a question that I have. I think it can be a generalist, but it's going to depend heavily on the genotype that we're looking at and the population. So it's not very simple, and I think I want to just go and target those specific populations where we're seeing genotypes reacting differently to evaluate what is happening. It might be that it's just something at local scales and not necessarily a broad tendency for the populations, sorry, for the species. And so why is all this relevant? What this is telling us is that when we're thinking about restoration or conservation for native legumes, it's very important to consider the native symbiont. So for instance, those different lupins that can be rare and actually endangered, and if you're trying to grow them and they are not growing, it could be because they need their specific symbiont. So the take-home message here is that for conservation and restoration efforts, it's very important to take into account these different abiotic relationships. And with that, I would like to thank to all the amazing undergrads that helped me to develop these different experiments. Of course, the members of the SACS lab and our collaborator, Stephanie Porter. And with that, I take some questions. Thank you.